Biological systems operate through a continuous, dense web of molecular interactions. Every cellular function relies on thousands of components interacting simultaneously. For decades, we have documented these processes using flat, textbook diagrams of metabolic pathways. But as this animation shows, a static map of the Krebs cycle completely changes when it bursts into motion, revealing a pulsing, dynamic network. The problem with a flat diagram is that it only maps the existence of a pathway. It tells us nothing about how it reacts over time, how it handles bottlenecks, or when external variables change. To understand cellular behavior, we have to translate these static biological drawings into functional mathematical models. This translational challenge is exactly what COPASI was built to solve. Short for Complex Pathway Simulator, it operates as a dedicated computational engine for systems biology. Developed by a collaborative network of scientists, it was built from the ground up as a free, open-source platform to ensure researchers everywhere had access to industrial-grade simulation tools. By converting structural biology into a computable format, COPASI serves as the critical bridge between theoretical biochemistry and the reality of experimental data. The software offers a streamlined environment designed to guide both novice students and experienced bioengineers through the modeling process. We are going to look at how to build a fully computable biological system entirely from scratch. The process begins here in a blank grid space. The first step is drawing a physical boundary box, which COPASI defines as a compartment. Without defining this physical volume, it is impossible to calculate accurate molecular concentrations. The physical space dictates the biological rules. Next, we populate that space. We introduce distinct molecular species, representing specific proteins or metabolites, directly into the compartment. Then, we connect these species by defining the biochemical reactions. A network of vectors links the molecules, establishing the pathway's flow. At this stage, we have successfully architected a structural map. However, this topology is purely physical. It still lacks the underlying mathematical rules required to simulate the passage of time. Knowing that molecule A converts into molecule B is useful, but a simple connection line cannot tell us the rate of that change. To simulate actual biology, we have to inject kinetic parameters. These are the mathematical rules that dictate exactly how fast each reaction occurs. In the software, real-world enzyme kinetics are mapped directly onto the reaction network. We overlay the vector arrows with specific mathematical operators. Researchers then configure these equations, setting specific values for variables like reaction velocity and enzyme concentration. Applying these specific values enables the model to accurately reflect complex regulatory mechanisms, like feedback loops or competitive inhibition. By assigning these kinetic parameters, we introduce a biological physics engine, effectively turning a static drawing into a computable simulation. With the architecture built and the mathematics defined, the model is ready to be set into motion. Looking at the graph on the left, we see deterministic simulation. This primary engine handles macroscopic systems with high molecular counts. It relies on ordinary differential equations to track average molecular populations over time, producing a perfectly smooth continuous output. But smooth averages fail when applied to microscopic systems. If a cell contains only a handful of specific RNA molecules, an equation built for millions yields inaccurate results. That is why COPASI provides a second engine, shown by the graph here on the right, stochastic simulation.
This is essential for modeling systems like gene regulation, where the molecule counts are extremely low. Instead of computing a clean average, the stochastic engine accounts for probabilistic noise, generating a jagged line that reflects the random microscopic fluctuations occurring inside a real cell. Both of these engines allow researchers to run time course simulations, giving them a window into how the biological system evolves second by second. Alternatively, researchers can run a steady-state analysis to bypass time entirely, evaluating how the biological network behaves once it reaches total equilibrium. Achieving biological accuracy is a matter of scale. You have to select the computational engine that mathematically matches the physical reality of the target system. Even with the correct engine selected, systems biology presents a harsh reality. and initial first-draft theoretical model almost never matches real-world observations. This drift happens because biology is messy. Initial assumptions carry unknown variables, and complex parameter interactions often behave unpredictably when scaled up. This graph visualizes the discrepancy. The ultimate ground truth is experimentally validated lab data, represented by these scatterplot dots. Right now, our theoretical solid line misses them completely. To correct this, COPASI employs a tool called parameter estimation. The software deploys optimization algorithms that run thousands of iterative loops. With every pass, it constantly tweaks and adjusts the underlying kinetic parameters, bending the math closer to reality. Eventually, the iterations resolve. The theoretical model forcefully aligns with the empirical data. Concurrently, researchers utilize COPASI's sensitivity analysis tool during this tuning phase. Sensitivity analysis systematically evaluates the entire network to identify exactly which specific parameters cause the most extreme changes to the system's final output. COPASI functions as an active reverse engineering engine designed to extract hidden biological truths out of incredibly messy datasets. A validated biological model loses its value if it remains trapped inside a proprietary software ecosystem. To prevent this isolation, COPASI is built with strict compliance to SBML, or the Systems Biology Markup Language. This standard translates the architecture into a universal format, guaranteeing it can be read by other platforms. Researchers can plug these SBML packages into bioinformatics pipelines using Python and R, integrate files into MATLAB, or upload them to the biomodels database. Strict interoperability ensures that a solitary simulation can expand into reproducible open science research. The models we construct today are laying the groundwork for how we approach human health and disease in the decades to come. Computational biology is moving from isolating single metabolic pathways to computing massive, multi-scale biological simulations that encompass entire organs. This scale paves the way for the digital twin, a functional digital replica of a patient's biological systems, providing a virtual proving ground for personalized medicine. As these models grow in complexity, the parameter estimation techniques required to balance them will increasingly rely on machine learning and AI-assisted analysis to process the immense volume of variables. Whether utilized by academic educators demonstrating enzyme kinetics or pharmaceutical researchers designing the next synthetic biologic, COPASI provides the necessary mathematical foundation. As the life sciences push further into the computational realm, open-source simulators will remain the critical engines we use to test the biological hypotheses of tomorrow.